We're gonna finish the nervous system. So this is our game plan. Right? If we make it a game, then it doesn't work. Game plan for Thursday. And I'm finding by integrating my keyboard, it actually gets a little less ugly up here. So like one, we're going to finish the nervous system. In particular, we need to do the ear. And once we do that, we are going to be done with all of the nervous system. If you have, we can kind of ask for any final questions about any of it. Then we're going to shift gears into the cardiovascular system. And for the cardiovascular system, we're going to look at blood. And we'll talk about what blood is made of and specifically the types of blood cells. And you are going to like talk about what they look like and how to recognize them. That could be something will be something like on your exams are looking at blood under a microscope and being able to identify what particular cell is at the pointer. And we'll see blood cells get stained with a special technique called Wright stain, and they have very distinctive looks, whether it's a lymphocyte or an eosinophil or a red blood cell or a macrophage or whatever. So blood, and then blood will be pretty easy. Then we're going to do the heart. So 2B, heart. And I can't ever write heart without putting a big, the heart. The heart is really, I was really kind of stoked that there was that heart model and that complete anatomy that I can use to kind of bring up as we're talking about it today. Usually what we do for the heart is we introduce the heart like today, then you spend time with the models and really get a sense of what they're like. And then the next class, we take sheep hearts and we dissect sheep hearts and really, obviously we're not gonna do that. But hopefully you will get a sense of the real kind of anatomy of the heart because, on a kind of schematic level, it's like, oh, the atria and sending out blood out the ventricles, out the aorta. But the reality, it's way kind of twisted in on itself. Like the blood comes in, but then the aorta actually has this valve and it comes out the very top. And it's all kind of wonky. So I'm going to do my very best using our virtual tools to give you a sense of, the heart, as an actual organ rather than just as kind of a functional pump. Because it's easy to understand it as a functional pump, but kind of looking at it and knowing which vessel is which and to orient yourself that way gets a little trickier, especially if it's not like red and blue and stuff like that. In a model, the aorta would be red because it's oxygenated blood. I should be careful. So that is our plan for today. We're going to finish the ear, introduce the cardiovascular system where we're going to kind of lay out the basic scheme of the whole thing, and then focus in on blood and the heart. And then next week, we'll go into the blood vessels and talk about the all the vasculature. And after that, that's going to be the division point for the first major exam. And the first major exam will probably be, let's see about this. Yeah, it's probably still going to be when it's officially scheduled for the 13th of April. And I think we'll still be able to make that happen. Obviously, it's going to be pretty different from the way we usually do it because the lab practical is going to be online. Um, there might be things even where we do some live thing where I can show you something on the screen and you have to answer something that you are looking at. I'm still trying to figure out exactly how it's going to look. I've got a couple of weeks to figure this one out. Um, so that is, you should keep that on your radar though, is that your next big exam is still going to happen. And all this studying for the quiz is going to be really helpful because that's, all material that you need to know for the exam. So let's see who's.
Wait, Professor, do you say April 13th? Because that looks like it's a Monday.
I wrote April. Maybe I spazzed on this thing. Let me take a look. I'm going into my calendar app. Ha! Yeah, I totally spazzed on my the official schedule I handed out to you. I wrote the 13th, but that was a mistake. So it's the 14th. Yeah, thank you for catching that.
Yeah, of course.
All right, so in fact, let me again, officially, I always feel like it's good to officially put it in writing. Exam 2, which is going to be both lecture, exam, and some version of a lab practical that we're going to figure out, is on April 14th. Again, if there is any reason that you think, because I know people's lives are kind of upside down and all that, if there's any reason you think you are not going to be able to be present for this kind of block of time on that day, please let me know as soon as possible so we can figure out how to approach that. But I do know that, again, with all the weird job stuff and kids out of school and all sorts of things, people's lives are pretty topsy-turvy right now. So we're trying our best to be flexible with things. Okay, let's dig in. So first, we're going to go over today's warm-up. Thank you, Elsa. I totally forgot. that was a good reminder there. So I'm going to share screen, and we're going to go to warm up to. I've been speed grading them. I don't think I've got through all of them, but after we're done with class today, I'll finish going through and grading them. And I am checking when people have typos and stuff. Again, it's a really fascinating thing. Like when you're trying to design a quiz for online, I can show you this just so you can, it's fascinating. It's like, I write a question about, what are these nuclei? And they're like, they're rods and cones. Except when you are trying to, some people, so I wrote rods and cones, and it's like, oh, maybe people write rods and cones, or the rods and cones, or the rods and cones, or rods, cones, or cones, rods, or cones and rods, or the cones and rods, or cones, and this did catch a lot, and I still missed it. I still miss people. And then obviously it doesn't understand if someone wrote cone forgot the E. So I'm still getting better, but it just means every time you if you want, yeah, this program, I wish it had a little more like AI to like figure out what a correct answer is from just the basics, but it doesn't. So let's go back and preview. And here we go. Fucking hell, there it is. All right, so name the overall structure indicated by the photo micrograph below. Most people got this, and it's a really nice view of the retina there. Again, here we can see that kind of pigmented layer. These are the rods and cones here. This is the kind of outer segment. These are the nuclei of the rods and cones. This is that plexiform layer where the rods and cones connect to the bipolar cells, which is this layer. And then this is the plexiform layer where the bipolar cells connect to the ganglion cells that make up the optic nerve. And the next question was, what kind of cells make up this nuclear layer indicated by this arrow? And those are the rods and cones. And so you kind of have to kind of orient yourself like, okay, I know I'm in the retina. This part here, this has got to be like the sclera, the choroid, and the pigmented layer. So that means these are the cones and rods. These are the bipolar cells. These are the ganglion cells. So that all makes sense? Yeah. Let's see. Where is everybody? There is everybody. All right, next we can go on. Name the structures indicated by the arrows in the photomicrograph below and give their function. So this, most people got that it's a taste bud. And these are kind of classic taste buds, these little things that look like, I don't know, I think they look like little onions sitting here. This is inside of a valate papilla. And in fact, let me add a different color so it's easier to see. You know, what kind of epithelium kind of lines the top of this stuff?
Stratified squamous.
That's all stratified squamous of your tongue. In your test, in your quiz that's coming up, as well as in all of your other tests, make sure that you answer the complete question. This had name the structures, which were taste buds, and give their function. So some people lost points. They only got 0.1 points because they wrote taste buds, but then they didn't take it farther. So you got to make sure that when you know, the classic thing that people say when you answer, you know, like answer the question and then go back and reread. This is supposed to be an A, that's supposed to be a D. Maybe I should do cursive. You know, reread the question after you've answered it and make sure that your answer is the correct answer for the question. You know, for a quiz, you know, a warm-up quiz, you lose 1/10 of a point. It's absolutely irrelevant. 1/10 of a point is not going to change your grade at all in any way, shape, or form. But if on a major exam, you do this a number of times, it can add up and it can actually make the difference between, you know, getting a higher or lower grade or a plus or a minus on something. So please be careful to pay attention to if it's a multiple part question. All right. I gotta go back to my mouse. There I go. Oh, I gotta clear out all this crap here. You know, for the following, choose cones, rods, both or neither. again, light energy into electrical signals. What is the answer?
Both.
It's going to be both. They are both phototransductors. That's the job they do, is they are doing the sensory transduction, converting light into electrical signals. High acuity.
Cones.
It's going to be cones. Cones are the ones that are the fovea and that can discern fine detail, whereas rods will tell you there's something there or not, but you can't really tell the details of it. You know there's a book in front of your face, but you can't read the writing in it. Which ones function best in bright light?
Cones.
Cones. Cones are also the ones that discriminate color. Rods are only monochrome, and rods are the ones that are allowing you to see stuff when the light is very dim. Are found in the retina.
Both.
Yeah, both. Where is different. In fact, here we have the cones are more in the center of the retina, really concentrated in the fovea centralis, the area right in the center of the retina. That is the place where the light lands when you stare right at something, the area of most acute vision. The rods, the rods tend to be on the periphery of your retina. Again, I don't know if I mentioned this, like if you were, here, oh, look, stars. There's some stars in the sky. And oh, there's the Big Dipper. If you were gonna try to look at a really faint star in the sky, why do they tell you don't look straight at it?
So the light goes to your cones.
Right, so if you stared straight at it, the light from the star would land on your cones. And if it's a very faint star, would you see it? Remember, cones only function in bright light.
No.
And so if you stare straight at these faint stars and they land on the fovea, which is where the light lands if you stare right at something, it's going to be mainly cones that are there, and they are not going to be able to respond to that very dim light. They need bright light to have a response. So if you look straight at a bright star, it just won't appear to you. Whereas if you look off to the side of if you look off to the side of the dim star, and then the light from that star lands on your rods, then all of a sudden you notice it, you can see it. It's really trippy. If you're ever just outside at night on a starry night, you'll notice like a dim star out of the side of your eye, and then you shift your gaze to look right at it. It disappears. It just winks out of existence. And then you look off to the side and it's like, oh, it's there again. And that's because the rods can detect the very dim light from the star, but the cones can't. Cones can't do the bright light. So stars, stars, stars. All right, let's clear away the stars and continue with this. What is meant by sensory transduction? Anybody?
Taking a stimulus and converting it to an electrical signal.
Exactly. So this is, taking a stimulus, I'll just use her words exactly, and converting it into an electrical signal, basically for the system to process. Right, that's kind of at the core of all of the stuff we've been talking about with the sensory division. Your retina, rods and cones, convert light into an electrical signal for your brain. Your taste buds, in fact, speaking of which, what is the function of the taste bud? I forgot to kind of finish that when we were talking about the taste buds. What is the function of the taste bud? Some people would just say, oh, the function is so you can taste. But what does it mean to taste? What does it mean to taste sweet? What does it mean?
detects different molecules.
Exactly. So when you really think about the function of the of tongue or of olfaction or gustation, function is to, you know, detect certain molecules. You know, in this case, you know, dissolved in your saliva. And when you're smelling, an olfaction is to detect molecules that are, you know, kind of floating around in the air. So chemoreception. But then once it detects, like for instance, sugar, it creates an electrical signal. That's the sensory transduction. Then the electrical signal goes down the nerve to, you know, the facial nerve or the glossopharyngeal nerve to your brain. So you know that that sugar molecule is in your mouth. This one here, I put this in here because we talked about, and I gave you little extra clues here to make it not sneaky. So you've seen this enough. So what are we looking at here? This is a photomicrograph of what part of your body?
The skin.
That's your skin. And here we've got the epidermis. And here we've got the dermis below that. And we have the little dermal papillae, those little papillae that go up. And then I made sure to make it clear that this is an encapsulated receptor. So you can kind of see that there's this little tic-tacky thing in there. And what are the encapsulated receptors that are sitting up in the dermal papillae?
Meissner's corpuscle.
Exactly. So that is a Meissner corpuscle, which responds to touch. So that, I thought that was kind of cool. Again, you can see it's there, you know, it's got this kind of more horizontal look to it compared to the more random swirly look of the loose connective tissue up in the dermis there, but it's pretty subtle. You know, in this picture with all sorts of clues, I had you, you know, I felt comfortable letting you recognize it, but I wouldn't just say like randomly, like, what is this thing? Because it's kind of subtle. So any questions about the warm-up for today? All right, so then we are going to move on and finish the ear. Okay. The ear. So I'll start just by saying that, you know, yes, it's responsible for hearing. And that's going to be this part called the cochlea. But it's also doing a lot of balance, equilibrium receptors. There's also a lot of equilibrium going on. There's going to be the 3 semicircular canals. These are for what we call dynamic equilibrium. There's going to be the saccule and the utricle. These are going to be what we call static equilibrium. So we are going to definitely look at the cochlea and hearing, but we're also going to be looking at the 3 semicircular canals, the saccule and the utricle. So the ear is a lot more than just the hearing. At the core of all of these is going to be mechanotransduction. Again, if you think about what is hearing even, hearing, you're listening to sound. Sound is air vibrations that are wiggling your eardrum. So ultimately, your ear is going to be translating movement of your eardrum into electrical signals. So at some level, you can even think about hearing as a version of touch. Something is basically kind of tapping and wiggling your eardrum. It's just, my vocal cords are creating these waves that tickle your ears inside and make your eardrums wiggle back and forth. But it's ultimately mechanotransduction, converting a physical movement into an electrical signal. Same thing when we look at these. It's going to be, as you move your head around, it's going to bend little things that get turned into electrical signals and send messages to your brain that your head is turning. At the core of all of these is this little mechanoreceptor cell called the hair cell. So I'm going to introduce the hair cell, and then we'll put it in the context into all sorts of different situations. So let me introduce to you the hair cell. It gets its name because it's got these extensions. There's one true cilia called the kinocilium, and then these other things that are actually stereocilia. It doesn't really matter. I always think it looks, I don't know, it looks kind of like Bart Simpson's hair or something. It's called a hair cell because it has these cilia and stereocilia coming up, which they call the hair bundle. The tallest of them all is called the kinocilium. The hair cell is also then going to be connected to a neuron that can send messages off to the nervous system. And the basic idea of a hair cell is it detects bending. And if you take that hair bundle and you bend it towards the kinocilium, that way, it becomes more positive, becomes electrically more positive. It's going to stimulate this more. It's going to send more action potentials down to its target. If you bend this thing in the other way, if you bend it away from the kinocilium, the whole cell becomes electrically more negative. And you'll have less. So this is a way for your brain to know which way this bundle is getting bent. If it's getting bent one way, the voltage is going up and it's sending lots of fast action potentials down the neuron. If you bend it the other way, it becomes more negative and the signal slows down. So depending on how we hook this up, we can use the hair cell to detect sound. We can use the hair cell to actually detect which way our head is spinning. We can even use the hair cell to tell us which way is gravity with respect to our head. So the hair cell is going to be at the core of this all. What I'm going to do now is, in order to kind of make things go faster, because I want to make sure we keep moving, I'm going to go into the Marib PowerPoint so I don't have to draw everything in this kind of wonky way. And I could just kind of show you these pictures. And all of these in fact, these PowerPoints are already on your Canvas. And the other ones we're going to do today with the cardiovascular system, I'll be putting up as well. So let's go. I need to go into, give me a few moments here. Marib PowerPoint, special senses. Okay, and open that and start that. And now we share. Okay. Wait, that did not, you don't see that. Wait, so you guys, this whole time, have you been looking? No, let me try this again. Just, new share. There we go. So let us move on here. The ear. All right, so things to pay attention to here. There are three parts of the ear. There is this external ear. The external ear is basically the part you see on the side of your head. The official word for that thing flapping on the side of your head is pinna. You should definitely know the word pinna. Helix is just that little ridge that goes around the edge there. The lobule is the earlobe. There's actually one other part which is not shown on this picture that I will show you later. If I don't get to it, please tell me to remind you, called the tragus. The tragus is actually like if you don't want to hear something and you push on that little kind of triangular thing on the front of your ear canal, that's called the tragus, but it doesn't matter so much. For our class, you don't need to know any more about the external ear other than what we're seeing here. There are some little muscles that can move it around. Non-human mammals have lots of crazy ability to move their ears around. I'm sure you've watched your cats and your dogs and stuff, put their ears up, put their ears down, listen to things, try to scoop the sound in. Our ears are usually, some people can wiggle their ears. I knew one person who spent a lot of time practicing isolating muscles and was able to do kind of amazing ear wiggling stuff, but that's not so typical. I'm not even sure a great use of your time. The ear canal, what bone is the ear canal going into here? If we take a look here, the ear canal is going in, this external acoustic meatus. What bone are we starting to tunnel into?
The temporal.
Yep, that's in the temporal bone. So everything else that we're going to talk about now, the middle ear and the inner ear, these are in the temporal bone. So they're in little caves in the temporal bone. In fact, we're going to get a little later, we'll talk about the bony labyrinth. So we're going to forget about the external ear now. We're going to focus in on the middle ear, which is, let me go to my pointer options again. Let's get the ink more light again. We're going to talk about this middle ear, and then we'll spend more time talking about the inner ear here. In general, the middle ear, this is where we find the eardrum. So the eardrum, this is the tympanic membrane. So tympanic membrane, also called the eardrum, that's the thing that vibrates when the sound hits your ear. The place where you actually convert the sound vibrations into electrical signals, that's going to happen in the inner ear. Or can I annotate with I can annotate with text. Holy crapola. Here we go. So actual sensory transduction occurs in inner ear using hair cells. So that is kind of important. The actual sensory transduction is going to be happening in the inner ear using hair cells. That being said, the inner ear is fluid filled. So the inner ear is fluid filled. Your sound is vibrations in the air. And if you, let me go back to annotate. Oh, hold on, I'm sorry. There we go. If I look at this whole system right in here, this middle ear is about taking the air vibrations and efficiently transmitting that vibrational energy into the inner ear where you're actually going to do the transduction. So these little bones, these are the tiniest little bones in your body. This is the hammer, also known as the malleus, the incus or anvil, the stapes or stirrup. You should know their names. You should know their names in English or in Latin as well as their common names. But they're basically going to take vibrations that will initially come and vibrate the tympanic membrane, the eardrum. Then these little bones act like a little lever system, which then push and pull and transmit that vibration into the inner ear. And the oval window is the little membrane that is actually being pushed and pulled by the stirrup in order to transmit that vibration into the inner ear. So we are going to spend most of our time looking inside the inner ear in a little bit, but let's start by looking at the middle ear because it's got some interesting stuff going on. Okay, let me clear all that crap off there. Okay. So middle ear, again, eardrum, also known as tympanic membrane, the ossicles, hammer, anvil, and stirrup, or malleus, incus, and stapes, and then they are going to be pushing and pulling on this little membrane called the oval window. You should definitely know the oval window. That's the little membrane so the vibration can get transmitted into the inner ear. Because the inner ear is filled with fluid, fluid is not compressible. So what happens is if you push on a fluid-filled environment, something else has to give. So you can't just have one window you push in on. You have to have another window then that pooches out. And that's what this little round window is down here. The round window basically can respond. If you push in on the oval window, then the round window is going to pooch out. If you pull back on the oval window, then the round window will have to suck in. So oval window, round window. Round window is just like the little release. So as you push and pull on the oval window, the round window will suck in the opposite direction. And again, that's going to be transmitting the vibrational energy into the inner fluid-filled or the fluid-filled world of the inner ear, which we'll get to in a few moments.
Are you going to draw this? Or should we be drawing it based off this picture?
Either way, you could draw it just based off. I thought this would be more efficient to just do it like this, but I could draw it as well.
Sometimes it's helpful for me, at least, when you draw it, because then it kind of streamlines exactly what we should be narrowing in on. But if it takes too much time, I understand too.
We could try. I mean, I can. It won't be as pretty, but it'll. So pay attention before I'm going to draw it. It's not going to be as pretty. I would pay attention to the fact that, so here is this middle ear, which is this little room inside your temporal bone. You know, one side is going, there's the inner ear, one side is the eardrum. You know, outside here is the air, whatever air pressure there is. If you don't have a way to equalize the air pressure in here, if you end up going, let's say up to Tahoe where the air pressure is lower and the air pressure in your ear was still the same as sea level, you would explode your eardrum outward. Because, or the other way, if you like dove to in a pool and the pressure starts getting stronger outside and you don't equalize and get equal pressure on the other side, your eardrum will implode. So we need some way to make sure that the air pressure inside the middle ear can equalize with whatever the air pressure is outside. And that is going to happen with this, what's called the, can be called the pharyngeotympanic tube, or the auditory tube, or a much more common word for it is actually the eustachian tube. And that is going to just go to the back of your throat, the pharynx, the nasopharynx, and it's a little hole basically up at the top of the back of your throat. And this basic, wow. You basically have this kind of, hey, you silly goose thing. Stop. Draw, there we go.
Is this what allows you to pop your ear?
Yeah, exactly, exactly. In fact, you can all do this. If you, again, this thing just comes out in the back of your throat up near your nasal cavity. So if you hold your nose, it's called the Valsalva maneuver, and blow, you're gonna feel your eardrums pooch out. So like everybody should do that. Hold your nose and blow out. Do you all feel your eardrums blow out? What other, did anybody feel anything else while you were at it? Any little tickle inside the inside part of your eye? Did anybody?
The eardrum starts to hurt if you blow too hard.
Say that again?
The eardrum starts to hurt if you blow too hard with your nose closed.
Yeah, no, you have to be careful. I also, when I do that, I start getting little bubbles coming out of my lacrimal puncta, right? Because my nasal cavity is also connected through the lacrimal sac into my, you know, inside of my eyes here. So I can actually feel like little bubbles happening inside my eyes when I do that as well. It's kind of weird.
So that tube allows you to equalize the air in that middle area.
Exactly. So basically you can take whatever the air pressure is out here and it's going in your mouth, in the back of your throat, and then it can come up and equalize in here as well. You know, and normally this tube is kind of stays shut, kind of floppy, but when you yawn or swallow, it kind of stretches it open and it allows things to equalize. A lot of times when people swallow or yawn is when like their ears pop and when everything equalizes. So I'm gonna just briefly here was the outer ear going into, again, now we're like in the temporal bone. Back. And again, that external acoustic meatus. And then you have, you know, the tympanic membrane. And this is going to be like the middle ear. Then that's going to go, you know, we're going to look at all of this later. It's going to go into the inner ear. And this is all in the temporal bone here. And again, those three little bones, the hammer, the anvil, and the little stirrup. And you want to remember that the little place that it actually pushes and pulls, those little bones push and pull on what's called the oval window. That is going to transmit the air vibration into the fluid vibration of the inner ear. And then we have this other one, the little round window, which is the thing that equalizes, round window. So as you push and pull on the oval window, this kind of can go back and forth the opposite way because the fluid is not compressible in here. So that's the basic idea. And then obviously I was talking about the eustachian tube, which is going to go down. Yeah, I don't think I can erase this properly. The eustachian tube is going to kind of go down and end up in the back of your throat to allow you to have air pressure so you can equalize the pressure in here versus out here. The other two things I'll add in, and I'll write their names down first, and then I will draw them on. There are a couple of little muscles here. Tensor.
I'm sorry, where would you equalize the pressure inside in the middle part of the ear?
Hold on, let me type this up and then I will get to you here. So say it again.
Where would you equalize the pressure in the somewhere in the middle ear?
Yeah, the middle ear, right here. You want to make sure your eardrum doesn't pop or explode. Like if you can't equalize that pressure, you will burst your eardrum. If there is enough of an unequal pressure on either side of the eardrum, it will either explode or implode. So like if you're really congested and you can't, if you're really inflamed here, this doesn't work, and you dive deeper and deeper and more pressure here and you can't equalize, you'll actually just implode your eardrum. You'll break your eardrum. So you can break the eardrum if the pressure does not equilibrate and it becomes more and more difference between the two sides. So tensor tympanum and stapedius are two little muscles in the inner ear, in the middle ear. Tensor tympanum, maybe I'll just do black. I haven't really used black. Tensor tympanum will actually grab onto the hammer to kind of tighten that up. Stapedius will actually grab onto the stapes muscle. And both of these, stapes bone, both of these muscles here, their job is basically to tighten up the bones in case the sounds get really loud. So it makes it so they can't move as much. So if you have really loud sounds coming in and you're worried you're going to damage your more delicate inner ear, you can tighten these muscles. It means these bones don't transmit the vibration so well, and you are protecting your inner ear if the sounds are really loud outside. So that's what these guys do, is they basically tighten up the bones if the sounds are really loud, so you don't transmit the vibration so effectively into the inner ear. So does that make sense? Yeah.
With those muscles, is that a subconscious thing?
Yeah, it's not something you control. In fact, I've heard even like in people who work in artillery or serve in artillery units and stuff, like before they would fire off like really loud shells, they would like fire off kind of a smaller shell first, which would then prepare people's, that would tighten up their inner ear. And so when the big shell went off after that, it was less likely to cause, damage to their ear. So it's something like when the loud sounds start, it starts happening automatically. Yeah, artillery is not, artillery can mess up your ears. Now we're going to go to the inner ear. So this is where the action's going to happen. This is where we're going to find the cochlea finishing the hearing. So far for hearing, air vibration coming in, vibrating the eardrum, transmitting through these little bones, vibrating the oval window. And then by magic, the hair cells are going to be converting that into an electrical signal, but we're going to look at that in more detail. But then we'll also see there's also the equilibrium senses in the inner ear here, the semicircular canals for rotational equilibrium, the sacral-neutrical for static equilibrium. So I'm going to go back to the PowerPoint because this is going to be way freaking prettier on a drawing that somebody prepared here. So inner ear, the middle ear, here on the middle ear again, this is just showing that tensor tympanum and stapedius muscle that can tighten up the bones so you don't transmit the sound so loudly to the inner ear. And next, this is going to be the inner ear. All right, so this is going to have lots of stuff going on. And in the interest of time, I probably will not be drawing all of this separately because I don't think I would do a better job than they're doing, and it would take a lot of time, especially on the iPad. So again, kind of going back, we started with the outer ear, the middle ear transmits the vibration through the little bones from the air vibration into this fluid world of the inner ear. So this is where we are now in the inner ear. Again, inner ear in this little cave inside the temporal bone. So here we're in a cave in the temporal bone. The cave has a name. The cave is called the bony labyrinth. So let's do the. So the cave of the bony labyrinth has the same shape as everything in here, but it's a cave. Like think about a shell of a snail. Like has anybody, if you've ever had escargot or something, basically the snail body has a snail shape, but it's exactly matches the spiral of the snail shell that it lives in. So you can think a lot about this as there's a shell that's actually a cave in the bone, and then this kind of membranous thing inside that fits in just perfectly. So the bony labyrinth is, let me use words for this. This is the cave in the temporal bone. That's kind of what I'm kind of drawing with all these kind of fuchsia arrows. It's like, it's hitting the boundary, but it's not, the inside is just hollow space, but then sitting inside the bony labyrinth is the membranous labyrinth. I want to get to my drawer again. There it is. Maybe I'll do it in green. All this stuff inside that's sitting in, that fits inside, all this green stuff is going to be what we call the membranous labyrinth. So somebody I think in their typing or something's making noise. If you could mute, that would be great. Thank you. Because otherwise, yeah, because this all gets recorded. All right. So the membranous labyrinth, I'll just put it over here, is basically all the stuff inside. that sits in there. For a lot of the things, for the equilibrium senses, we're not really going to care about so much the bony labyrinth part. But when we get to the cochlea, the sense of hearing, you actually do need to think about the bony labyrinth versus the membranous labyrinth. But for, oh my God, look at the time. To start here, I'm gonna go hopefully finish up this ear in the next 10 minutes here. For the semicircular canals, which do your sense of rotational equilibrium, like which direction is your head spinning? And for the utricle and saccule, we can just think of them in terms of the membranous labyrinth and think about the fluid in there, the fluid which is the endolymph, and think about the hair cells and how the hair cells are connected up. So let's start with the well, let's see what this thing goes to. Let's not do that first. Let's start with this, the saccule and the utricle. So here in this part of the membranous labyrinth here, we call this the vestibule in here. So inside the vestibule is where you're going to find the utricle and the saccule. These are going to be telling you which way does gravity pull. A lot of times when in fact, these other things here, these semicircular canals, even though they're not officially in the vestibule, we usually consider them also quote unquote vestibular organs. When people talk about vestibular senses, so these are going to be your equilibrium senses. These are not about hearing. This is going to be about, in terms of saccule and utricle, which way is gravity going? Or in terms of the semicircular canals, which way is my head spinning? For the saccule and the utricle, they are exactly the same thing, just at different orientations, like perpendicular, so they can tell different directions. But the basic idea is, in fact, we'll come back here. I'll go to, hold on. So again, these are sitting inside the vestibule in the inner ear. At the core is going to be hair cells. We talked about hair cells being the main things on all of our inner ear. So hair cells. And again, the hair bundle detects bending. The voltage changes when it bends one direction or the other. On top of that is going to be this big gelatinous thing with these little calcium carbonate crystals. They're called otoliths, means ear stones. You know, you can kind of think of them as like little rocks. So in your inner ear, you've got these hair cells sitting in this little patch. We call the little patch the macula. Macula just means like spot. And they basically got this thing with rocks sitting on top. So if you bend your head, the rocks are going to get pulled in one direction or another by the force of gravity. And as they do that, they're going to be bending the hair cells they're sitting on. And as the hair cell bend, they give the brain information that you are, moving your head with respect to gravity. So if we kind of go back to this drawing here you can see really nicely, again, the hair cells, the little otoliths on top. We have our total groovy looking person here. Like, I wonder who does their hair. Leaning forward. As they do that, the little rocks are going to get pulled due to gravity, which is going to bend the hair cells. And that's going to send a message down the neuron to your brain that your head is tilted with respect to gravity. Does that make sense? Yeah. And if we're looking at the utricle or the saccule, it's the same basic idea. It's just they're at different orientations to each other, but they're both the same basic organ. So that is, and I can also say, go back. They're also called, sometimes they're called the utriculus and the sacculus. So saccule or sacculus is the same thing.
So the utricle and the sacculae, those make up the rocky sac, or is that correct?
No, this whole organ is called the saccule or the utricle, including the macula and the otoliths on top. Together, this is, it's either the utricle or the saccule, depending on which one you're looking at. They both have the same basic structure.
So they both have the hair cells and the rocks.
Yes. This thing you're looking at here is, I wish they didn't call them two different names. You could just call them, with the semicircular canals, you're just going to say, oh, we have 3 semicircular canals with different, in different orientations. These are the same basic idea. They're the same organ, but in different orientations. So these could be called the vestibular patches, but they're not. They're called the utricle and the saccule.
So do those line the entire semicircular canal?
Say it again?
Do those, are they throughout those canals, or is there?
No, they're not in the canals at all.
Okay.
No, they are here in the vestibule. One of them is here, one of them is here. Do you see that?
Yeah, I get it now.
Yeah, so those two are in the vestibule. Now what we're going to talk about are the semicircular canals, these things that look like these big loops coming off of the vestibule. There's 3 of them. They just have 3 different orientations. They're for rotation, so you've got, roll, pitch, and yaw. So I can go on to, can do this really briefly. Let's go to share content, whiteboard. Yes. So there are 3 semicircular canals. And they're basically, so you can tell, the 3 different rotational axes. And each one basically looks like this. It's got a semicircular canal. It's got this swelling called the ampulla. It's filled with fluid. Endolymph is the fluid in there. And then in the ampulla, you have a little organ called the crista ampullaris. which is basically going to be some hair cells. With this gelatinous membrane on top called the cupula. The gelatinous curtain. So the basic idea here, so and again, together, these two things equal the crista ampullaris. The basic idea here is the endolymph, this fluid has inertia, and everything else, this is all your skull, right? These are all caves in your skull, right? This is all temporal bone. So if you move your head, if you like spin your head one way or another, this whole bone is going to be spinning. This whole thing is spinning, but the fluid has inertia. You know, this is where it probably makes sense to let's see, we try to do, give me a second here. This will probably work. I'm gonna turn off my background so you can see this better. So if I have something heavy, it has inertia. I have something underneath it. If I pull fast, we have, you know, the inertia. This thing stayed still, but this thing moved with respect to it, right? If I move this slowly, it follows along. But if I, you know, inertia, you know, Choco Cat stays still, but so think about this being the skull. The skull is moving. This white paper is the skull. Choco Cat here is the endolymph, the fluid that doesn't move. So the skull is moving, but the endolymph is not moving. And then you can think of air. The water in the ear is also just a fluid, like air is a fluid. Let's say this was that little cupula, that little membrane. If I go this way real fast, then the air pushes it and this thing bends that way. If I go this way real fast, then this bends the opposite direction. So whichever way I'm pushing this whole structure, this little floppy membrane is getting bent the opposite direction. So if we go back, actually, could you see me in that little corner there? Well enough?
Yes.
Yeah.
Okay, so let me go back to my, so if we go and look here, somebody, let me erase a lot of this just to like,
well been like, oh,
so now we can say, all right, somebody is spinning their head like this, which means this little thing is trying to go this way, but the fluid here is staying still. It's going to bend the cupula backward. The cupula is going to bend. And when the cupula bends, the hair cell is going to bend, and that's going to send a message to your brain that you are spinning. So that makes sense?
Can you explain it one more time with like what the curtain's doing?
So this cupula, this little gelatinous thing here is floppy. So it can bend. So if I am spinning my head, this thing is kind of attached to this whole structure, but the fluid is not moving. The fluid has inertia. So as this structure is rotating, it hits this fluid with the inertia, and then that actually causes this thing to bend backwards and bend the hair cells. Does that make sense?
Yeah, thank you.
So I'm like looking at the, hold on. It's 2 o'clock. Let's take a break because it is 2 o'clock. And let's come back at 2:40, and we'll like wrap up the ear, and then introduce the heart. And then we'll also talk about some lab stuff and all that. So any questions about this before we take a quick little break?
What was the cupula ampullaris again? Was it that structure with the curtain?
Yeah, so the crista ampullaris is just the little hair cells and that cupula on top that's sitting there in the ampulla. So it's kind of the official name for that little organ there.
And then once the cupula bends, is that what signals to your brain that your head is turned?
Exactly. Right, so as I turn my head one way or another, it makes the cupula bend, and that's how my brain knows that my head is bending. Is because the cupula getting pushed by the endolymph bends the hair cells, and that's the thing that converts movement to electrical signals.
Thank you.
Sure. All right, so actually now so let's say 2:45. 2:45, we will be back here and continue. So we got the saccule and the utricle. Both of these are in the vestibule. And these do what we call static equilibrium. It's basically which way is gravity pulling. Sometimes people think of these as linear acceleration. Because if you're like, if you like hit the gas pedal on your sports car, you're going faster and faster and faster. It's the same thing as if gravity's pulling backwards. So these saccule and the utricle are also going to be responding. So you can talk about static equilibrium. Even if you're standing still, it tells you which way is gravity pulling. That's why static, you're not moving, but it's still giving you important information. Then there were the semicircular canals. These are kind of coming off of the vestibule, but often they're included when we talk about the vestibular organs, or the organs that do equilibrium. These are doing what we call dynamic equilibrium. Sometimes called rotational acceleration. So like the rotational acceleration of your head, or also dynamic equilibrium, what's going on as your head is moving. Dynamic, if things are constant, then it's not responding anymore. So I can do, sure, I'll do this for you. Oh, for this, it's useful. So if I need to turn off the background again. Again, my home life is a little ratty here, but so if I am standing up and and I start spinning, as I spin more and more and more and more, I'm actually not really getting dizzy. I could keep talking to you. I can keep doing this. I'm not going to fall over. I'm actually feeling just fine. why is it right now that I don't really feel dizzy, even though I mean, I see the world spinning around weird. Why do I not feel dizzy?
Because you haven't stopped.
Because at this point, the fluid, the endolymph, is moving along with my skull. So as long as the fluid is turning and my skull is moving at the same way, same rate, then the cupula doesn't bend and there's no signal to my brain that anything is off. If I stop all of a sudden, I don't know, my eyes are probably going crazy. Right now, I have to hold on so I don't fall on my face or don't barf. Why do you get dizzy if you're spinning around and you stop?
The cupula is bending.
And why is it bending?
Because the fluid has inertia.
Exactly. The fluid has inertia. The fluid is keeping moving, even though your head isn't. So the fluid is hitting onto the cupula and bending it and telling your brain that you're spinning, even though you're not. So you're going, whoa, I feel like I'm turning, but I'm not turning. And everything gets all funny until the fluid kind of stops moving and everything's back to normal. So let's get back to background. All right. To finish up, we need to talk about the cochlea, where you actually have actual hearing happening. And again, in the interest of time, normally I spend a lot more time on the cochlea. I like hearing a lot, but I really also want to make sure we get through all the material here. So we'll get into the real, just most important parts of what's going on in the cochlea. So I'm going to go back to the PowerPoints here. And here you can see, this is showing the cupula and the semicircular canal as this fluid is moving relative to this, bends the cupula and bends the hair cells, sends a message to your brain that, again, if it's everything's straight, you feel like you're not spinning. But if the head is moving with respect to the endolymph, cupula bends, sends a message to your brain, you feel like you're spinning around. This is showing the cupula and the hair cells, and this whole thing is called the crista ampullaris. And so now let's get to the ear. Back. Let's go, what happened to the ear? There it is. So the basic idea here is the vibrations that are the air vibrations come in into your main ear canal and vibrate the eardrum back and forth, which is then going to transmit that vibration through the ossicles, those three little bones, onto this oval window. And that's going to be setting up these pressure waves in this fluid, in this endolymph, but actually not in the endolymph. So the membranous labyrinth is going through the bony labyrinth. And then there's the bone, and there's little canals that all, in fact, let me go back. Yeah, sorry, I was trying to skip some steps to make it quicker and easier, but didn't help. So, back. Clear. So here is the membranous labyrinth, this blue part, spiraling around in this part called the cochlea. Cochlea means snail. It looks kind of like a snail. It's got 2.5 turns. So 2.5 spirals going in. So membranous labyrinth spiraling in 2.5 times, and it's spiraling in the cave. That's the bony labyrinth. So the bony labyrinth in the whoa. So as the bony labyrinth kind of makes the outside, the membranous labyrinth spirals inside, you end up getting 3 separate little ducts. So the membranous labyrinth creates what's called the cochlear duct. But then above and below it, you're going to have the vestibular duct and the tympanic duct. So here, if you like look at, the snail, this is kind of going in and out of the board here. For every rotation, you can have 3 different ducts. You have this upper duct, the vestibular duct, you have this middle duct, the cochlear duct, and the lower one, which is the tympanic duct. And to make our life easy, I'm going to just show it unfolded. But before we do that, I want to add one other piece in here. In this cochlear duct, in the middlemost of these 3 different channels, that is where you're going to find this spiral organ of Corti. And let me give it let me put my text tool here. So spiral organ, often called the organ of Corti. And this has hair cells with an overlying, what they call tectorial membrane. If you remember, the hair cells are the things that actually, are going to, I'm trying to move this thing, but I don't know if I can. Maybe I can't. So when you have vibrations in this fluid world, this is ultimately where the vibration is going to get converted into an electrical signal. This organ of Corti, this is kind of showing it in magnification with little hair cells in here that are going to, and again, we don't have time to go into the outer versus inner hair cells because I want to keep moving. But let's just leave it that there are hair cells here in the organ of Corti that can convert vibration into electrical signals. And so now, I'm going to take a look here. As the vibration kind of enters into the inner ear, as the oval window gets vibrated, the vibration initially goes into this vestibular duct, goes all the way around. The very tip of the snail is called the helicotrema. This is the tip of the snail. And then it kind of comes back. We talked about how that vibration or that pressure wave gets released at the round window. And as that vibration is going through, ultimately it's going to vibrate this membrane here underneath that cochlear duct. So again, if I go back, this is that cross section of that spiral going up. The vibration starts going in the scale of vestibuli or vestibular duct, goes all the way out to the helicotremas, coming back along this tympanic duct. As the tympanic duct vibrates, this basilar membrane vibrates. And as this basilar membrane vibrates, the hair cells on the organ of Corti are going to shake, and that is going to send a message to your brain that there's vibration, there's sound. the spiral ganglion. What is a ganglion?
Cell body in the PNS.
Exactly. These are actually the cell bodies of cells. I think they're bipolar cells that actually connect to the hair cells and go on to create cranial nerve number what?
8.
Exactly, the 8th nerve. So this is the cross section of the cochlea. If I go back here, this is showing it kind of unwound and big. And the basic idea, again, I've got all this crap on here. Basic idea, the vibration goes in the vestibular duct. Helicotrema is the end of the tip of the snail comes back. The vibration vibrates this basilar membrane, which then sends a message to, or which wiggles the hair cells, which sends a message to the brain. The other thing you need to know here is this, let me get the drawer again. The basilar membrane, closer to the helicotrema, this vibrates best, it absorbs low frequencies. So that's going to be important, actually. The basilar membrane up near the beginning here, this absorbs high frequencies. Let me. You can have things that just have like a mechanical resonance. It's like a guitar. You have like the low E string is kind of heavier and thicker, and its natural vibration is a lower frequency. The high E string is much tighter, and its natural vibration is much higher. You know, that frequency that it optimally vibrates at is just due to the mechanical properties of the string. It's the same thing in your basilar membrane in your inner ear here. The low frequencies are maximally going to be absorbed and shake the basilar membrane, and therefore the hair cells on top over here. High frequencies are going to maximally shake the basilar membrane, and therefore the hair cells in the organ of Corti over here. So this is how your brain can get information about the frequency of the sound. Does that make sense? Kind of, sort of?
So it's just in the different parts of the basilar membrane, they'll pick up on the low frequency or the high frequency, but it's still just within the basilar membrane, correct?
Correct. So like over here, if I erase clear drawings, you know, here it would be, you know, areas more around here are going to be absorbing higher frequencies. Here's the helicotrema. Up here is absorbing lower frequencies and stimulating hair cells that are up here. So as you spiral 2.5 times around, it's optimally going to respond to frequencies going, you know, lowest at the very tip here, highest at the beginning here. That makes sense? And then again, if we take a cross section through this thing, what it ends up looking like, you basically, you know, you come in, you cross the in and out and in and out and in and out as you go round and round. So you're going to end up having, looks like this. Again, there's 3 ducts around and around and around. Under the microscope, I'm going to show you some pictures under the microscope. Hold on 1 second. Give me 2 seconds here. anatomy, images. Oh, that's not it. Hold on. Hold on, sorry about that. I am still going to, I thought I had it there, but I have it someplace else. Photographs. Histology. Here we go. Okay, I am now going to share my screen here. New share. Oh, no, it's, do you see it? Do you all see the photo micrograph?
Yes.
All right, so that is a photo micrograph, actually from our own collection of one of those little turns in a guinea pig cochlea. It's small enough, we can show it here. So this is basically, this is the cochlear duct here. This is the tympanic duct here. This is the vestibular duct here. This is the basilar membrane. This is the vestibular membrane. This is actually that little tectorial membrane sitting on top. These are the hair cells. The organ of Corti is right in here. Capital because it's named after a dude. So this is kind of what it looks like under the microscope. Over here, I don't know if that looks familiar to you. Maybe let me see if I can zoom. Come on, zoom. Maybe not. Oh, here, so you can see, let me clear here. Again, here we are still in the cochlea. And here we've got the basilar membrane, the organ of Corti. This is the spiral ganglion. This is actually the cell bodies of the neurons that make up the cochlear nerve. Again, the cochlear nerve meets up with the nerves from the vestibule and the semicircular canals to create the vestibulocochlear nerve. Now you know why it's called vestibulocochlear nerve. It's called vestibulocochlear nerve because you are getting stuff from the vestibule and from the cochlea. So I know I've kind of blasted through this. But are there any questions about the ear here? All right.
What specifically do you want us to know? Like the vestibular membrane, the cochlear membrane, or the vestibular duct, cochlear, and tympanic, and then
you should know that so you vibrate. In fact, maybe let me put in a text window here. You should know kind of the basic idea that, that vibration comes in. You have some, sound. Oh my God. Sound basically equals kind of an air vibration. This is going to vibrate the tympanic membrane. That is going to, ossicles, then vibration to oval window where where vibration enters inner ear. Then in the inner ear, vibration initially goes up the vestibular duct and returns down the tympanic duct. In this process, the basilar membrane, again, which divides the cochlear duct, and the tympanic duct. Tympanic duct will vibrate. Location depends on frequency. And higher frequency up near the down closer to the oval window, low frequency down up near the helicotrema, and shake the organ of Corti. And as hair cells bend, they create an electrical signal which will travel to the brain via cranial nerve. Ta-da. So does that kind of make it clear?
Yeah, that does. Just to clarify, is the receptor the actual hair cell, or would you say the whole?
The organ of Corti is kind of like, think about taste bud. A taste bud is the little receptor organ, but it's the little gustatory cell that does the actual transduction. The organ of Corti is like the part of the inner ear that's, the business end of converting vibration into electrical signal, but the part of it that's doing the real converting movement to electrical signal is the hair cell.
Okay, thank you.
Sure. All righty then. So then we should any last questions regarding obviously if you have other questions later, it's not your last chance ever to ask a question. But does anybody have any questions before we're going to move on to the heart and the cardiovascular system? Okay, let me look at the time. It's 3:14. It's like pie time, 3.14. I think what I'm going to do, I'm going to introduce the cardiovascular system, give you kind of the basic background, and we might take one more break before we continue on into really getting into the details of the heart. So let us do this then. I'm going to So we always start a new system by talking about kind of functions. So what are some of the important functions of the cardiovascular system?
Pump blood.
Say it again.
To pump blood.
So I would say pumping blood is the mechanism that it accomplishes its job, but why do we pump blood? What's the function? What's the purpose of sending blood all around the body?
Circulate oxygen.
Yeah, so a big part of it is going to be kind of delivery of oxygen, of nutrients. What other kinds of things does it deliver?
Removes waste.
So it's gonna, so delivery of, I'm gonna put hormones in here, but also kind of gets rid of, takes away,
Autumn.
wastes, carbon dioxide, nitrogenous waste like urea. What are other functions of the cardiovascular system?
Fights disease. Like it keeps out all the foreign particles out of the body.
So we're going to say staging area for the immune system. Your immune system is actually all over your body. The white blood cells, when we talk about white blood cells a little later, we'll see, yes, there are some inside your bloodstream, but there are lots of them that are not in the bloodstream. They're in the lymph, they're in lymph nodes, they're just actually moving around in the connective tissue spaces. So, but that being said, there are a lot of blood cells that are part of your immune system that are inside your bloodstream and use it as a way to travel from one place to another, and can actually, it's called diapedesis, like your white blood cells sense that there's something going on that they need to attend to. They'll actually grab onto the wall of the blood vessel and squeeze out and leave this closed circulatory system and go into the tissue space to start dealing with the infection. So staging area for immune system. There's a lot of important parts of your immune system that have like a home base in the cardiovascular system. Other things.
To help regulate temperature?
Yeah, totally.
And like pH fluids.
That's going to be more the urinary system. The urinary system does pH. Cardiovascular, though, is going to definitely be part of your temperature regulation. You can change the amount of blood going to the surface of your skin by like 50 times. So if you really need to cool off, you can send 50 times as much blood to the surface of your skin to try to radiate out heat and cool off than when you're cold and you try to keep the blood closer to the core of your body. So temperature regulation will be another part. So all of these things, and delivery of oxygen, because, you need to bring oxygen to very close to every cell in your body. Oxygen does not diffuse all that far, all that fast. If you want the oxygen to get from the blood to the cell, the blood vessel has to be really close to the cell, like a fraction of a millimeter. We're going to have somewhere about like 100,000 kilometers of blood vessels. That's like 60,000 miles, if you want to think about it in terms of miles. So you just have blood vessels everywhere, getting really close to every part of your body to deliver oxygen, nutrients, take away wastes, and all that. So what we're going to start is just looking at the big picture, how this whole thing is laid out, and then we'll look at each part individually. But we'll start first with the big picture. So we'll put in the heart. So I'm drawing a heart looking like a heart here, just so you know it's the heart. But the reality is the heart is really not that simple when you start looking at it realistically. But I think to start out explaining how it works, it's good to use this really super simplified picture of it. And then we will transition. I'm going to do initially talking about here are the basic circuits of blood flow, the basic chambers, but then I'm going to change and make it much more realistic. But so this is an unrealistic, that's why I'm drawing it looking like a Valentine's thing, because I don't, to make it really clear, I'm going to draw this in a way that like the blood vessels are coming out totally the wrong place. So we're going to have a right and a left side to the heart. Both sides are constantly, with every heartbeat, both sides are pumps that are sending blood out to one circuit or another. They just work in conjunction. We have the ventricles and the atria. So like this here, this is going to be my, left atrium. This over here is going to be my left ventricle. On the other side, it's going to be the opposite. This is going to be the right atrium and the right ventricle. The ventricle, this is like the main pumping chamber. When you think about the heart, the atria is kind of where blood is returning to the heart, and it kind of basically flows through. And it's the ventricles that are the ones that squeeze to really generate the blood pressure and push the blood out into the body. When the ventricles squeeze, you want to make sure that the blood, so we're going to have these blood vessels, these big arteries that are leaving. When you squeeze these ventricles and the blood leaves, so any vessel where the blood leaves the body is called an artery. Oh, leave the heart. And, for completeness, when we have blood returning to the heart, what's going on? Let's make it blue. When it comes back to the heart, it's going to be deoxygenated. Anything that's returning to the heart is going to be a vein. It's important to get away from some of your old assumptions about arteries and veins being red and blue. We're going to see veins that are red. The veins that are returning back from the heart, from the lungs after you've picked up oxygen, they're going to be bright red because they have just picked up oxygen, but they're veins because they're returning to the heart. When you have arteries carrying the blood to the lungs to pick up oxygen, that blood is blue, but it's still an artery. That's the pulmonary artery coming out there. So arteries are defined by leaving the heart. They don't leave the heart, they leave the heart. Or lead away, they leave the heart. Veins are always returning to the heart. Doesn't matter if they're red or blue. Other things we can add into this picture, when the ventricles squeeze, we want to make sure the blood keeps going out to the body or to the lungs and does not go back up to where it just came from. So we're going to have valves. They're called the AV valves or atrioventricular valves. These little one-way valves, kind of like swinging saloon doors. You can swing them in, but you can't, once they close, you can't go back the other way. So if I take a look here, this is going to be an atrioventricular valve on my left side of the heart. On this side, I'm going to have an atrioventricular valve on the right side of my heart. Both of them, again, so you can't have blood going back from the ventricles up to the atrium when the ventricles are really squeezing. The names are this. So the left atrioventricular valve is called the bicuspid valve. Or also called mitral valve. If we go on the other side, the right AV valve, so they get their names based on how many flaps they got. The bicuspid valve, yes? Bicuspid valve, if you actually looked at this thing, it would have like these two flaps, kind of like two barn doors. They could swing if you're going through the right way, but then they come back and they don't open. So bicuspid, meaning two cusps, two flaps. It's called mitral valve because it looks like the hat on a bishop. The bishop wears a hat called a mitre, and he's here and he's saying like, "Be gone, coronavirus, I command thee," or whatever. They've got these hats that look like that. So that's why they call it the bicuspid valve, or they call it a mitral valve. But please, Elsa, watch out there. It's because of the similarity to a bishop's mitre. Tricuspid valve, if you look at it, it has three cusps. So that's why it's called the tricuspid valve. That's why they get their name. But their function is exactly the same. Keep blood from coming back from the ventricles back up into the atrium when the ventricles contract. While we're here, while you're sending blood out of these major vessels out into the body from the ventricles, like here we're sending deoxygenated blood to the lungs. Here we're sending blood out to the body. It's like in this case, this would actually be my like aorta. The aorta is the main artery that leaves the left ventricle going out to the body. This side is going to be what's called my pulmonary trunk. It's the main artery that leaves the ventricle and goes off to the lungs. When the ventricles relax, so here's my left ventricle, my right ventricle. When the ventricles relax, you want to make sure that the blood, which is now under quite a bit of pressure, doesn't come back into the heart. So we have another set of valves called the semilunar valves at the entrance to these major arteries leaving the heart. So let me erase the Pope here. And got aortic and pulmonary semilunar valves to prevent backflow back into ventricles. Oh, cool. All right, so here we got one here and we got one there. So prevent blood from flowing back from the pulmonary arteries back into the heart. One over here to prevent blood from flowing from the aorta back into the heart. One of the things, these actually are also made with little flaps. They have 3 cusps. Oh my God. The reason why you need to make sure you remember that is the right AV valve also had 3 cusps. So if you're looking at a picture or a real heart or a model or something, and you see 3 flaps on the valve, it doesn't mean it's the tricuspid valve. It might be one of these puppies. So that's why you want to be careful to, like, just because, you know, yes, bicuspid valve has 2 flaps, tricuspid valve has 3 flaps, but these semilunar valves also have 3 flaps. So don't use the number of flaps as the thing that tells you definitively, oh, I know what valve I'm looking at. So now what I'm going to do is put this heart in a bigger picture of the circulation. And basically, that main artery that leaves the, oh my gosh, that main artery there that is leaving the left ventricle, again, which is my aorta, that is going to send blood everywhere to every little nook and cranny of your body. Ultimately, it's going to branch into smaller and smaller little things until we get into what are called capillary beds. We'll talk about more, but they're basically simple squamous epithelium tubes where the nutrients and oxygen and everything can diffuse out of the bloodstream and into the tissues. In general, if a blood vessel is containing blood that has a lot of oxygen in it, we draw it as red. Blood that has attached a lot of oxygen to the hemoglobin becomes bright red. When the hemoglobin releases some of its oxygen, it becomes darker colored. It doesn't turn blue, but through your skin, it looks bluish. And so the convention is, you know, after we have delivered oxygen to the body, then we have the veins that are bringing the blood back to the heart, that we draw them in blue. The blue means that we now don't have as much oxygen in the blood. You know, the blood that's returning to the heart here, to the, this is my right atrium. This is going to be my pulmonary veins. I mean, I'm stuff. This, the vena cava. This is the, in fact, maybe I should draw it as 2 things. It's actually the superior and inferior vena cava. Let me erase you, little puppy. There we go. So let me, maybe I should type it even. It'll be prettier. Normally it would be like superior vena cava, inferior vena cava, but 2 of them are vena cavae. So basically, the superior vena cava is draining all the blood from the upper part of your body. The inferior vena cava is coming up, draining from your toes up to the lower part of your body. So those are the major veins that are returning to the heart from the body. We call this side the systemic circuit. Because the blood is kind of going out to the whole body system and then coming back. Then once it's on the right side, that deoxygenated blood is going to get pushed out to the lungs. This main hose that comes out of the right ventricle that's carrying the deoxygenated blood to the lungs, this dude is called the pulmonary trunk. You know, which divides into kind of right and left pulmonary arteries. So again, remember, these are my pulmonary arteries. They're blue. So please never think that artery's got to be red. This is pulmonary artery, goes blue, goes up, goes to the lungs. Again, we're going to have capillaries as well, where we can exchange material, absorb oxygen from the lungs. Then we can bring it back, return into the heart here. You know, this is going to be our pulmonary veins. And again, red veins because they are carrying oxygenated blood back from the lungs. And then I can add in this side here is called the pulmonary circuit. So this is kind of your basic main circuits. There's a couple of other circuits you need to know. The heart muscle itself is incredibly metabolically hungry. It needs a lot of oxygen to keep pumping. And you also need to make sure that it gets its supply or else nothing else. It's kind of like, you know, the plane's going down, the oxygen mask drops, you put the oxygen mask on yourself, so then you can help the other people. So the blood supply that's coming out of the aorta, there's an immediate tap that goes back into the heart to supply the heart muscle itself. So there are these arteries that come straight off of the base of the aorta and go and supply the heart itself. And what are those called? Does anybody know the name of the arteries that directly supply the heart muscle?
Coronary artery.
Exactly, the coronary arteries. So as soon as the blood is pushed out, there's a tap, the right and left coronary arteries that supply the heart muscle itself, so the heart has what it needs to keep on pumping. One other thing I don't know if this is going to work. Hold on a sec. Whoa, that's not what I meant to do. Well, let's see. Let me try this different way. One other thing I can show you that's important. We have this circuit going to the body. Circuit going to the lungs. We have the coronary circulation that's actually going directly into the heart itself. And then there's one other circuit or type of circuit that you need to know, and it's called a portal system. So both of these, the systemic circuit and this pulmonary circuit, is kind of like arteries go out into the capillary bed, And then the capillaries drain into veins that go straight back to the heart. Same thing on the pulmonary side. You've got pulmonary arteries going to the lungs and to the capillaries of the lungs. And then the capillary bed drains and comes straight back into the heart. One example of a portal system is in your digestive tract. Like when you absorb your nutrients from your food, that is going to be picked up by the bloodstream. So for instance, you're going to have arteries that go and feed capillary beds in your digestive organs. So you go send blood to the digestive organs, absorbs the nutrients, but you don't want to put it directly into the bloodstream first. The next place it goes actually is it goes to the liver, and it goes to a second capillary bed sitting in the liver. Usually I draw this in purple, so I don't have purple. Pretend this is purple. This is going to go into a second capillary bed. Trying to like make it so you can actually read it. So second capillary bed in the liver. So portal system, this, in fact, what I've just drawn here is called the hepatic portal system. Maybe I can erase this other stuff to make it all more clear. Basically, capillary bed 1 drains into capillary bed 2 and then returns to the heart. Right? Because normally you go out to the capillaries, then you come back to the heart. In this case, we actually go first to the digestive organs, pick up nutrients and such, but then we take the blood and it goes into a second capillary bed where it's processed in the liver. The liver can store things, it can detoxify things, transform things. So it's not until the absorbed stuff from your digestive tract has been processed by the liver that it actually finally returns, to the heart here. So does that make sense? There are going to be other portal systems that we're going to see before the semester ends. We're going to see the hepatic portal system right here, but we'll see the hypophyseal portal system when we do the endocrine system. Actually, in the nephron of the kidneys, we're going to see another version of a portal system with the glomerulus and the peritubular capillaries. So this isn't the only example of a portal system, but it's a really super famous one. And it's one that is going to play a role in our discussion of the cardiovascular system right now, because we're going to talk about the hepatic portal vein that's draining out of the digestive organs to the liver, but then the hepatic vein, which drains the liver and goes back to the heart. So you need to make sure you kind of understand this idea that you can have a vein that actually is draining into another organ rather than back to the heart. In this case, in my drawing here, this thing right here, this is what we would call the hepatic portal vein. You know, it's a vein that is leaving the capillary bed and on its way, but it's still got a stop to make before the blood is actually going back to the heart. So any questions about that? Yes, no? Okay. So let's take a 15-minute break, and I'm going to think about the best way to have you guys start thinking about the heart. Actually, before I do that, though, let me give you just a little preview. Like this picture that I've been drawing that makes it look, oh, I see the blood is coming into the atrium, leaving the ventricle, going off to the body. You know, the reality is this semilunar valve here is actually up here, and the aorta is coming right out the top of the heart. And this semilunar valve is also up here, and the pulmonary trunk is coming out right off the front of the heart here. And everything is all kind of twisted around. So actually understanding what you're looking at gets a little trickier in reality, but I think it's useful to think about this functionally like this to start with. And when we come back, I'm going to kind of orient you more realistically about the heart and hopefully actually have just more hands-on things for you to work on rather than me just keep talking to you. So let's take a break, and let's say at 4 o'clock we will regroup. All right, so the real heart does not look like a Valentine's. It looks kind of more like this. It's got like this apex. This is actually called the base. It does have those 4 different chambers, although the ventricles are really obvious. The atria are really kind of floppy and not all that well defined because they don't have to hold a lot of pressure. The left ventricle has really thick walls because it has to squeeze really hard to generate high pressure to send blood all over to the whole systemic circulation. You know, if we kind of go back, I don't have it anymore. You know, the right ventricle is much thinner walled. Like when you're trying to orient yourself looking at a heart that's been cut open, the ventricle that has really thick walls, it's always going to be the left ventricle. Also, it's not all totally symmetrical. The left ventricle kind of goes more down to the bottom here. And again, we'll look at this in more detail in a few moments. So these AV valves are indeed kind of right here. This is like the bicuspid and tricuspid valve. But the aortic semilunar valve and pulmonary semilunar valve are also at that same midline. The pulmonary semilunar valve is here. The aortic semilunar valve is here. Actually, it's a little behind it. So like the pulmonary trunk is coming out right up the front. Paul Morney? Good God. In fact, the main vessel that you see when you first look at a heart, if you're looking at it head on, it's the pulmonary trunk. The aorta is got its aortic semilunar valve behind. It's coming up right behind it. and making this big arch. It's going to be my aortic arch here. So that's one of the big ways it's different from what I've been drawing for you. Another thing is the atria have these little expansion things. They're called auricles. Auricle means ear, and they look kind of like ears. So maybe I'll use yellow here. Let's say this is my right ventricle, not right atrium. There is this little thing that just kind of expands and makes this little extra expansion pouch that makes this little weird little sac-like thing that looks like an ear on the heart. You have the same thing on the other side with the left one. So we have auricle on this side and auricle on that side. So when you look at the heart and wonder, what are those? That's what they are. When you are looking at the heart, I want you to make sure you think about the blood flow. A very common question is not just what chamber are you in, but what is going on? Is the blood oxygenated, deoxygenated? Where was the blood before it got there? Where is it going after? I am very fond of test questions that deal with, it might be Benny blood drop. Or could be like little Debbie drop-o blood. And little Benny or little Debbie are going to be, maybe little Debbie is in the vena cava. Where did she end up next? And you got to know that, oh, the vena cava is going to return, oh my God, into the right atrium. And, as we get more complete with all the blood vessels, it could be little Benny is in your thumb and has to return back to the heart and go out to the lungs and get more oxygen and come back. So it's going to get more and more complicated. But for right now, just think about if little Benny or Debbie is in one of these chambers, where were they right before? Where are they going next? What kind of valves do they need to go through to get there? So just kind of think about it functionally as well as anatomically. So now I'm going to just go and do a lightning round with the PowerPoint. And here we go. Lightning round with PowerPoint. Starting. And here you just see those circuits that we talked about. This should all be pretty straightforward to you all now. Again, you should look at this and make sure you really think about what the flow is. You are going to think about it functionally as well as anatomically. Aorta going out to the body, returning to the left, to the right atrium, and then from the right atrium through the tricuspid valve into the right ventricle and out through the pulmonary semilunar valve, up the pulmonary trunk into the right and left pulmonary arteries to the lungs, coming back into the pulmonary veins. Here you see another reality check. The pulmonary veins, there's actually 4 of them. There's 2 coming in from the right and 2 coming in from the left, all showing up in the left atrium. So then from there, you can go out to the body, la, la, la. All right, let's continue. La, la, la. We got to get rid of clear drawing. We're going to talk more about the situation of the heart next week. It's in the pericardial sac, which is important to talk about. But for right now, let's just talk about the sections of the heart. Oh, and here, just from looking at this, you can see those auricles I'm talking about. Auricle, again, comes from the word for ear. And it's because if you look at these kind of weird things here that look kind of like ears on the heart, those are actually the auricles. This is the right auricle and the left auricle. And again, they look like there's something special, but they're just the kind of expansion chambers to allow for more filling in the atria. This is kind of giving you a sense of how the heart is also not how I was talking about. You know, this is that apex of the heart. So like this is all part of the left ventricle. The right ventricle doesn't start till kind of like up here. So the right ventricle is kind of smushed off on the side. The left ventricle kind of takes up the main position. And you'll see when we slice it, it's really clear that that's happening there. Again, this is kind of, you should be familiar with looking at the heart from the front like this. You should be able to, this is what it looks like if you're looking straight from the front. That's how I kind of orient myself is you've got this pulmonary trunk. It's this main thing coming up. This, didn't mean to do that. This is going to be leaving through from the right ventricle out to the lungs. Behind it is the aorta coming up from the left ventricle, making this arch, the aortic arch. We're going to learn about all the vessels coming off it a little later. The vena cava, those are the ones that were returning from the body back into the right atrium. There's one big one coming in from the top, draining your head and your arms. And then there's another one coming up from the bottom, draining your lower body. Those big vena cava are the ones that are draining the systemic circuit. You should be able to recognize those kind of where they are on the heart. Pulmonary veins coming in, draining the lungs. Again, they're red because the blood coming back from the lungs has oxygen. There's 4 of them. They're all going into the right atrium. This is showing if you cut all, or you carve away all the fat and everything. Here we have the coronary arteries. There's the right coronary artery and the left coronary artery, which kind of comes out the other side. Kind of going down this sulcus here between the right and left ventricles. So that's that. This is from the back. Here you can see, you know, the pulmonary trunk is actually in front, so you don't actually see the pulmonary trunk, but then you can see the right and left pulmonary arteries coming out. You can see the aorta arching out, superior, inferior vena cava. Here are those 4 pulmonary veins coming into the left atrium. This thing, the coronary sinus, you should know the coronary sinus. We talked about the coronary arteries are the things that are supplying oxygen to the heart. And the drainage of those vessels is all these coronary veins. All the coronary veins ultimately drain into this coronary sinus. This is just where all the blood returns from the coronary circulation and just dumps back into where? In fact, the blood that's returning that has delivered oxygen to the heart muscle itself and is now coming back into the heart, where is it going to come back into? All the blood, the deoxygenated blood that comes back into the heart is coming into where?
The right atrium?
Say that again?
Is it the right atrium?
Exactly. So it drains directly into the right atrium. Perfect. So, So here is the heart that's cut open. You should definitely be familiar with this kind of view of the heart. Here you can see the left ventricle has really frickin' beefy walls. That is a dead giveaway that this is the left ventricle, because it has these really beefy muscular walls, because it needs to be able to generate a lot of pressure to keep the blood flowing throughout the entire systemic circulation. The right ventricle, which is more pushed off on the side here, and has these, this kind of wimpier looking wall. So just by looking at the size of the walls, you get a sense of which is the left, which is the right. Here you can see the atria are not so well defined. They're kind of floppier bags where the blood just kind of returns in, and with these AV valves, atrioventricular valves, leading into the ventricles. But in general, the atria, it's not like the blood all fills up the atria and then goes into the ventricles. It's like as the blood is returning into the body, it basically flows through the atria and starts filling the ventricle. By the time the heartbeat starts, only like 15% of blood is left in here to get joined into here. Most of the blood is already in the ventricle. It's just been pouring through. So it's not like the blood goes into the atria and then gets transferred into the ventricle. It's more, it pours through the atria into the ventricle. And right at the beginning of the heartbeat, any extra blood that's still in the atria gets squished into the ventricle. So that's why they're really not that well defined. They're kind of just a place, they squeeze to dump last bits of blood into the ventricle. This is showing when it's not colored with red and blue. You can still see that pulmonary trunk is this big hose, rubbery hose coming out the very top. The aorta is this big arching rubbery hose behind it. The veins tend to be floppier. Here's the vena cava, which is behind there. Clear. And this is the same picture. Eventually, you should be able to look at a heart like this and know what you're looking at. I would like you to recognize, the pulmonary trunk, the aorta, not just when they're colored bright blue and bright red, but from their basic form that you would see on a nicely dissected real heart like this. So again, it gets trickier when you don't have the color to help you out. So that's why I think starting with the pictures and the colors and the models, but then we will eventually transition to being able to recognize it when you don't have the colors as well. Again, the colors make it way freaking easier. You should be able to look at this. And again, the left ventricle, like I said, takes up more of the heart than the right. And this little sulcus between the two ventricles, this is where you have one of the coronary, this branch off the left coronary artery coming down here. This is, I think, the one that they usually call like the widowmaker, the one that often gets blocked during in heart attacks. And, again, a heart attack is just a coronary block of the coronary arteries. And if those coronary arteries are blocked, you are not going to get blood to your heart muscle to keep it alive. And that's a heart attack. So you don't want that. Other things, when you're looking at the heart in more detail, again, make sure you recognize the different valves. And again, you should be able to know that this is the bicuspid valve because it's on the left side. And this is a tricuspid valve because it's on the right side leading into the right ventricle. This is the pulmonary semilunar valve going from the right ventricle up into the pulmonary trunk. This little bitty bop here is the little bit of the aortic semilunar valve that would be heading up into the aorta from the left ventricle. Other things that are cool here. Let me clear my drawings. Things that you will be able to look at and study in a few moments. So we have the valve here, and it's under a lot of pressure when the ventricle's beating. So you want to be able to kind of reinforce it. And there are these little strings that hold and keep the valves from blowing backwards. They are called the chordae tendineae. Here's the word for them right over here. You should definitely know chordae tendineae. And the chordae tendineae are anchored on these little muscles that are in the ventricle. They're called the papillary muscles. And basically the papillary muscles contract and they pull, and they basically pull on those little chordae tendineae in order to make sure the valves don't blow back under pressure. I think Marib sometimes says, oh, the chordae tendineae are like your little heartstrings. So those are your little heartstrings, the chordae tendineae. Other things that you can see in here. The meat, the lining of the heart is really kind of textured. That weird texture that you see in there are called the trabeculae carnae. It means like the I think the crossbars of meat, basically, because when you look at the actual heart muscle, it's not smooth in there. It's all got these kind of lines and grooves and stuff. That's all trabeculae carnae means. It's all those lines and grooves. Continuing on our whirlwind tour of the heart. It's kind of looking dissection of the heart. This is actually pretty cool. This is kind of showing that midline of the heart where all the valves are. I was saying that the tricuspid valve, the mitral valve, and the semilunar valve from both the pulmonary and aortic, they're all on the same level. They're all in this kind of area right between the atria and the ventricle. They're all kind of reinforced with connective tissue. We're going to come back to that. The fibrous skeleton of the heart reinforcing the valves as well as being important for the electrical activity. Let us continue. Whirlwind tour. This is again showing how all the valves are actually kind of all next to each other, as opposed to how I drew them on that first picture in the very beginning. Oh, this is kind of, if you were on that little submarine with Raquel Welch in The Incredible Journey, you would see this as you go through the heart. These are the little heartstrings, chordae tendineae, anchoring the flaps of the AV valves with these papillary muscles to kind of pull and hold, make sure that during the contraction, the valves are reinforced. This is showing a real heart. If we were cutting up our sheep hearts, you'd see this. Again, it's real easy to know where you are because you look and you say, oh my God, here is a chamber with really freaking thick walls. This has got to be the left ventricle. And on this side has got to be the right ventricle. And here's a little papillary muscle and the chordae tendineae going onto the valves. So again, thinking about the thickness and orienting, eventually, you know, by 2 weeks from now, I would hope if you looked at this picture, you would be able to know which is which ventricle or which chamber is which. So when you're starting to think about this all and orienting yourself, start with the pictures that have color and are more kind of, you know, easy to grok, but then move on to stuff like this. See if you can start orienting yourself on real hearts and things that aren't colored. This is just the whole flow. Again, make sure you think about the flow. Blood's returning through the vena cava into the right atrium. From the right atrium goes through the tricuspid valve into the right ventricle. From there, through the semilunar valve into the pulmonary trunk and the pulmonary arteries out to the lungs. You should definitely be, that should just feel like, oh yeah, I know that, I got the flow. Your exams will most definitely follow, again, it might be Bernardo blood drop or whoever, but he's going to take a journey that's definitely going to be going through the chambers of the heart as well as lots of other things. We'll do all the rest of this later. Well, this is just showing those coronary arteries coming right off the base of the aorta, the left and the right coronary arteries. And again, they just go straight to supply the heart muscle itself. The drainage of those ultimately all come into the coronary sinus, which then is going to dump into the right atrium. And let us go share. So, can you all see this beating heart?
Yes.
So this is, again, this would be what the heart looks like. It's a simulation, it's a model, but this is kind of what it looks like here. You know, again, coming, if I look at like here, this is going to be the left ventricle, which kind of extends down to the apex, the right ventricle here. And we know also the right ventricle, it's heading up here. Here's the pulmonary trunk going out to the lungs. The left ventricle is going to be connected to the aorta going out to the body. These are the little auricles. This is representing the superior and inferior vena cava, draining the head and arms versus draining the lower body. Over here, these are the pulmonary veins, the veins that are coming back from the lungs going into the left atrium. So this is the left atrium right here, which then is going to dump into the left ventricle, which is this one, which then is going to spoosh out into the aorta, which is here, and go off out into the body. And again, I am going to, as soon as I log off here, what I'm going to do is first is upload all of the Merib PowerPoints for you. I have to rename some stuff. And then I'm going to do my very best to make sure that you all can get free versions of this software as soon as possible, because I think it's really useful to, when you're thinking about, in fact, there, that little hole there, that is actually supposed to represent where the coronary artery is leaving. There's going to be one on that side and another one down in here. Right, so you're going to have the right coronary artery coming off and the left coronary artery coming off. So let's stop sharing. So I just threw a lot of stuff at you. And again, this is non-ideal, but I'm thinking it's probably going to be more useful for you to know what you're trying to recognize on the heart and spend some time actually just, and if you want, I'm